Okay, our next speaker, Tom Theeuwen, is going to talk to us about the genetics of limitations and how to identify when photosynthesis limits crop yield. Your title is the same this time? Yeah, I got caught out once already. I thought I'd better check. Thank you.
No, it's completely fine. Thank you very much. Good afternoon, everyone. I'm a research fellow at the IngenHousz Institute, an institute that most of you probably haven't heard of about yet, but that's because it's a new institute. It's only a year old. It's based here in Wageningen, and it works basically on photosynthetic research. I'm establishing my own group on genetics of limitations, and in this talk, I'll want to focus on, together with you, on the question, how do we identify photosynthetic limits for crop yields? But first, why photosynthesis? I think plant breeding over the last, well, thousands of years, but let's be a bit more recent. The last few decades have done an amazing job on keeping improving yield and yield components. Though, if we look at the formula for yield potential, and it's basically comprised of radiation interception efficiency, conversion efficiency, and harvest indexed. And primarily the advances made are in radiation interception efficiency and harvest index. And there is probably still room for some improvement, but the improvements are getting closer and closer to the optimal. And the one thing that's left out at this point is the conversion efficiency. And this is effectively photosynthesis and photorespiration. And this is where we come in and we want to see whether we can do something about this. Because, simply said, photosynthesis and yield don't correlate easily, because otherwise we would have already fixed it. Why is that? Here we have a nice illustration of the light reactions of photosynthesis. Is a very simplified version of it, but you can already get a bit of an idea of how complicated it already is. The light reactions produce NADPH and ATP, and this is being fed into this carbon-benzene cycle, and photorespiration, which all are interacting with via the stomata and depend on those as well. And I'm just giving you this simple illustration, but you should realize that this is all based on roughly 3,000 genes that take control of this. So it's quite a complicated process, and this is just on the leaf level. And this is all based on the genotype and the environment, and those interact and perform all of these photostatic processes together. So this is already quite a complicated interaction. Now, based on the genotype and the environment and the interaction of those, it depends on how this photosynthesis is then translated into yield. So if a plant breeder is just focusing on yield, it's not very likely that that all of the photostatic processes are already optimized. And so let's have a little closer look at this. So let's start disentangling this a little bit. This brings me back to my PhD work, which I was doing in Arabidopsis. And I was phenotyping, in this case, 7 different accessions, doesn't really matter. And I was looking at photoprotection, one of the many traits involved in photosynthesis, and there was some variation there. Okay, cool. But I was interested in why is this one very much different from another one? And I produced a cross, I made doubled haploid population, I made a decent sized population to have a proper statistical power. And the interesting thing is where it came that we started using the DEPI chamber. These are chambers in Michigan State University. And the great thing about this is A, that we can have light intensity fluctuate, and B, the fact that the ceiling is full of cameras, chlorophyll fluorescence imagers, and that means that all of these plants inside can be phenotyped simultaneously. And so we can simultaneously, at all of these instances, these are 5.5 days, have an idea how photosynthesis is doing in response to these environmental conditions. So if we then start looking at a specific time point for this trait, photoprotection/MPQ, and we start dissecting the genetic architecture, we see that already here is like 4, possibly 5 QTLs here, which was already more than expected because these two traits, okay, they differ a little bit, but I had not expected it to be so different. If I then slightly flip the graph around here, you can see the same data as here, and everything below the threshold are grayed out, and all the peaks are still there. So these are the 4 QTLs in a different orientation. And why I'm doing this? because if you now look on the whole time scale, these 4 QTLs that were present there in the beginning, some of them disappear, some of them are more persistent, some of them differ, there's new ones coming up. But basically what it means that it depends entirely over time and thereby environment on how the genetic composition makes the phenotype. So this is actually quite complicated already. But why you can also see is that I have color-coded these either greenish or bluish. And this means that I call it this due to the parent effect. And that means that any blue QTLs have a phenotypic effect that is caused by one of the parental alleles, and the green effect is caused by the other parental allele. Okay, so I think it's safe to say that there is a plethora of genetic variation for just one trait, but in reality, we already had a look, like photosynthesis is not one trait, but many traits. And it's a highly dynamic environment, this, but I only changed the light intensity. In reality, the environment is much more complicated than just this. And let's say, okay, all of that we can do. Then the last thing, and it's probably even a more daunting task, okay, we have all of this variation, but which of these alleles for these QTLs is actually important for improving photosynthesis? Because that highly depends on the environmental conditions, and not an easy question to answer. So I think that the answer to this is that a certain allele for phenotypic variation for photosynthesis only matters in light of yield. And we can look at this crop, wheat in this case, grown throughout the whole growing cycle. You can see in the beginning it may be exposed to frosty conditions, then very sunny conditions, flooding even, or just wet, dry quite right after, sunny, and then it's being harvested. At all of these stages, all of these photostatic processes are happening. And it's the sum of all of these things that in the end up making yield. But it may well be that there is only in specific time points that photosynthesis is actually limiting. And in others, it may not. So it's very hard to just by selecting on yield to see this. So what we are now hoping to do is to actually start measuring photosynthesis throughout all of this time and then determine how this contributes. And so that brings me to the problem definition. I think it's safe to say that there's ample of genetic variation for photosynthesis, though there is a strong genotype by environment by time interaction. And that makes it too complicated or too complex to improve substantially based on selection on yield purely. And even if we were able to do so, if we were able to measure all of the underlying photostatic processes, it's very difficult to tell whether a certain allele is beneficial or not. So that's why we want to measure photosynthesis throughout time in the field and build a platform, build a method to determine when a certain variant for photostatic variation actually is limiting yield. That's all very great, but how do we do this, right? That's a nice dream. Here you can see two devices that are typically being used to measure certain processes of photosynthesis, a so-called spectrophotometer, a chlorophyll fluorescence imager. These work great in measuring photosynthesis, but they're not really suitable to have in the field to monitor a segregating population of whatever crop throughout the whole year. That's why we at the JAI have developed now this little sensor that's on my hand that combines the capabilities of both, and they're actually quite cheap, and that means that we can put them like this on a leaf, and then not just one, and also not 10 or 100, but 1,000 in a field, and then have them monitor photostatic processes, in this case chlorophyll fluorescence. That would look something like this. So you could imagine that this is now continuously measuring photostatic processes, and then get a certain data stream. But that's only more focusing on the light reactions. I think most of you will probably agree that sort of these gas exchange systems are probably the golden truth in doing photostatic measurements. They're quite expensive and not very high throughput, let alone being able to do this on a whole year. So we are now dreaming out loud whether we can even have one of these on a little chip like this. All right. That's now this. Let's produce some time series. So we're very much at the prototype, but we have some working versions at the moment. My colleague Ching Cheng had 3 of these sensors as a trial out in the field. And the interesting thing is this is not only measuring photostatic processes, it's also its own little weather station. These are quite closely together placed in a crop. You can see it measures temperature and light intensity, and while at some points certain light flecks are present, you can see that it's quite different. It quite differs between these sensors, so it's already quite complicated. So it's not that one environment is determining the whole field. No, it's much more complicated than that. And the plant is responding to that using photosynthesis. And we measure this fluorescence trace, which you can use to measure roughly 15 parameters, depending on how you do it. And that gives us an idea of how these plants are doing. This is already complicated, but I remember that I just said, like it only matters what photosynthesis is doing in light of yield. And yield is obviously an endpoint measurement. But if we say, okay, let's look at biomass instead, we can actually see how biomass would grow using high-throughput phenotyping systems that we've heard about quite a bit over the last 2 days. And then it's a matter of seeing whether we can correlate, or correlate is probably not doing very much justice, but whether we can see patterns there that are underlying this. So let's go back to what I just showed you. We have now the genotype information. We have a very good idea of the environmental variation. We know quite a bit about photostatic variation over time in the field. And we have the yield/biomass information. The part that we're missing is this green box, not the black box, but the green box. And that's the interaction. And this is the part which we want to model. I put a simplified version up here. The first step in this is to identify and train a model. And this is a very visionary point at this point. We're working on the first baby steps in here, and we've heard quite a lot of people talk about this also the last few days. And we want to be open-minded in this point and try a range of different models at this point. This can be dynamical models, statistical models, machine learning models, AI models, or a combination of these in terms of hybrid models. At the moment, we are exploring ODE models together with Sjoerd Boersma up there. And we are seeing whether we can use these to capture this interaction. But we're also exploring statistical models, in this case splines with Martin Boer up there as well. But that's all great, because that's only the part for the data to the crop modeling. But I think when it really becomes interesting is when we bring this to the knowledge. And that's when we are extracting the model, extracting knowledge from the model. And while this is just a short list of options we have, we are now thinking whether we can use things like feature analysis, sensitivity analysis, and whether we can use QTL analysis to extract knowledge from this model. And you can think about something like this. It's a very simple image. Like for each parameter that we measured using these sensors, which we measure over time, so let's say this is sowing and this is harvesting, which is, bear with me, that's not what is the graph really showing, but you could realize that a certain trait at some point is more important than in other traits. So the importance of a certain trait at a certain time point would tell us something about when this particular trait is limiting. And that's a nice step, because that brings us to the third step, because we've now identified a photostatic limitation, but that's just a hypothesis, right? Because we don't know whether this is true until we start testing this. And this is where genomic selection comes in, and this is something that we're exploring together with Louise and Jonathan as well. So there's a lot of this variation. A lot of it is small, and we can now use, like, okay, let's say, oh, maybe these ones are important and other ones aren't. So then we develop a genomic selection approach and then use speed breeding to quickly bring that all together and bring it into the field and test it whether we were true, whether this was really a limiting factor. Now, the outcome it can be at that point, well, we were right, okay, but then probably it means that there is another limitation again, so that brings us back. It could also be that we're wrong and we have to go back to the drawing board. So you can see a vicious cycle appearing there. And that brings me already to the end of it. I want to thank the IngenHousz Institute, also the people from Biometris, and my supervisors for my PhD. And please reach out if you have any ideas or in this regard. Thank you very much.
Thank you, Tom. Take one question if we have one.
Hi, thank you. So first off, I don't really know anything about photosynthesis, so this may be a stupid question. But that little device you had on the leaf, and it does this laser thing or whatever on the leaf, and it does it probably a lot of times during the day and during the growing season. Does that influence the photosynthesis on that specific spot in the leaf by repeatedly measuring it?
Yes, it's a good question, a very good question even. Yes, it does. So that will be the limiting factor in how often you can measure. In theory, the hardware would be there that we could, in theory, do this every second. But at some point, you have to find a balance in not affecting the plant or that particular spot and the resolution of the data. So that we should find a balance there. So, yeah, that depends a little.
Okay, thank you.
Thank you very much,
I'm happy
Tom.
it was not stupid, the question.
No, it wasn't. Definitely not.
Okay, thank you very much. I think we'll close it out there. And it gives me the opportunity to introduce our final invited speaker.